Welcome to Weekly Neurology Deep Dive, produced by America Gavanini. Today we're taking a close look, a real deep dive into some fascinating research published in the Annals of Neurology. The paper is titled Blood-Synuclein Separates Parkinson's Disease from Dementia with Lewy Bodies. And it really gets into some novel ways we might be able to distinguish these conditions using blood biomarkers. Just a quick reminder before we jump in, this discussion is purely for educational purposes. Think of it as a summary of the published study. We definitely recommend reading the full paper for all the details. This isn't medical advice, and there are no commercial interests here. Okay, so for those of you needing the main points right away, here's the quick version. This study suggests that different strains or shapes of alpha-synuclein in blood plasma might help tell Parkinson's disease apart from dementia with Lewy bodies. They found plasma levels of two specific strains called strain A and strain B were significantly higher in Parkinson's disease compared to dementia with Lewy bodies. This held true across two large groups of patients. And importantly, for those with Parkinson's, higher levels of strain A at the start predicted a slower decline in cognition later on. These alpha-synuclein bits in the plasma also seem capable of causing more protein aggregation in lab tests. So this points towards potential blood tests for diagnosis and prognosis, maybe even hints at disease mechanisms outside the brain. Now, let's unpack the problem this research tackles. Lewy body diseases that includes Parkinson's and dementia with Lewy bodies affect, over 10 million people globally. The hallmark is these clumps of misfolded alpha-synuclein protein inside neurons, the Lewy bodies. But telling them apart clinically can be tough.
It really can. Clinically, we diagnose Parkinson's based on the motor symptoms, the slowness, tremor, stiffness. And dementia with Lewy bodies, that's characterized by dementia appearing before or within a year of the motor symptoms. But here's the kicker. When we look at the brain pathology after death, Parkinson's disease dementia and dementia with Lewy bodies look almost identical, same diffuse Lewy body spread. So we're missing reliable diagnostic biomarkers to separate them during life. That's a major gap for us.
And it raises this fundamental question, doesn't it? Why does pathology that looks so similar under the microscope cause such different symptoms while someone is alive? Plus, cognitive decline is a huge issue. It eventually affects about 70% of people with Parkinson's, hitting quality of life hard. And we know this cognitive decline tends to correlate with Lewy bodies spreading into the cortex. So figuring out what drives alpha-synuclein misfolding and spread is, it's critical. Now, scientists have known for a while that alpha-synuclein isn't just one thing. It can fold into different shapes or strains. Think of it like different versions of the same protein, each potentially behaving differently. This study uses some new tools to ask, could these strains be the key differentiator we've been looking for?
That's the core idea. The researchers developed specific tools, monoclonal antibodies called 71 9A027. These antibodies were designed to recognize two distinct alpha-synuclein species they created in the lab, which they termed strain A and strain B. And these strains seem to behave differently. Strain A mainly seeds more alpha-synuclein clumps. Strain B, interestingly, seems to pull both tau and alpha-synuclein into clumps, and it might get cleared out more easily. So the main aim was to use these specialized antibodies to measure these specific alpha-synuclein species. They looked in human brain tissue, in cerebrospinal fluid, and crucially in plasma. Samples came from people with Lewy body diseases and also from neurologically normal individuals. They used techniques like ELISAs for measuring levels, Western blots, immunohistochemistry to visualize things in tissue. Importantly, they also used seed amplification assays and cell models to see if these strains isolated from people could actually, cause the disease process.
Okay, so a multi-pronged approach using these novel antibodies. The big question then is, did it work? Can these antibodies actually help distinguish Parkinson's from dementia with Lewy bodies?
Well, one of the first really surprising findings was where they didn't find much. In the cerebrospinal fluid from people with Parkinson's disease, levels of these strain A and strain B alpha-synuclein species were actually very low, almost undetectable. This held true even when they pooled cerebrospinal fluid samples from multiple individuals with Lewy body diseases. So these specific strains don't seem abundant there. But then they looked in the plasma, and it was a completely different story. In plasma from the same groups of patients with Lewy body diseases, they found robust, easily measurable levels of both strain A and strain B alpha-synuclein. This was really striking, especially given how accessible plasma is compared to cerebrospinal fluid.
Right, that contrast between cerebrospinal fluid and plasma is fascinating. But did the plasma levels reflect what was going on in the brain? You might assume they would.
That's what you might think, but no. They looked for correlations between the plasma levels of these strains, or total alpha-synuclein, and the levels found in brain tissue samples or seen with brain staining. and they found no significant correlation. This really suggests that the alpha-synuclein species they're measuring in the plasma might not just be leaking out from the brain. It raises the possibility of a source outside the central nervous system, or perhaps they're packaged differently, maybe in extracellular vesicles.
Okay, so plasma seems to be a unique window. Let's get to the core clinical challenge, differentiating Parkinson's disease and dementia with Lewy bodies. How did the plasma strains perform there?
This is where it gets really interesting for clinical practice. In their first cohort from the University of Pennsylvania with around 235 samples, they found that plasma from individuals with Parkinson's disease had significantly higher levels of alpha-synuclein detected by both the strain A and strain B alleles. This difference was clear when comparing them to people with dementia with Lewy bodies, but also compared to neurologically normal controls and even people with Alzheimer's disease. And naturally, they needed to see if this would replicate. So they tested it in a second independent cohort. This was a multi-site group from the NIH Parkinson's Disease Biomarker Project, about 200 samples. And again, plasma from the Parkinson's disease group showed significantly higher levels of strain B alpha-synuclein compared to the dementia with Lewy bodies group. Strain A levels were also higher in Parkinson's in this cohort, though the difference didn't quite reach statistical significance in this group. The p-value was just over 0.05. But when they used statistical methods, these receiver operating characteristic curves, to see how well the plasma levels could distinguish the groups. They confirmed that adding the plasma alpha-synuclein strain measurements significantly improved the ability to tell dementia with Lewy bodies apart from Parkinson's disease, compared to just using age and sex. And this was true in both the University of Pennsylvania cohort and the multi-site NIH cohort. That replication is key.
That does sound powerful. Finding a consistent difference in a readily accessible fluid like plasma across two independent, well-characterized cohorts, that's potentially a very big deal for diagnosis. Okay, so it helps with diagnosis. What about prognosis? Can these plasma markers tell us anything about how someone's condition might evolve, particularly with cognition?
Yes, they looked into that as well. They had follow-up data on about 95 individuals with Parkinson's disease, and they found something quite specific. Higher baseline plasma levels of strain A predicted a slower rate of future cognitive decline. This association was statistically significant, with the p-value less than 0.01. So higher strain A seemed protective against rapid cognitive worsening. Interestingly, plasma strain B levels didn't show this relationship with cognitive decline, and neither strain A nor strain B predicted the rate of motor symptom progression. So it points to a specific link between plasma strain A and the trajectory of cognitive function in Parkinson's disease.
That's really intriguing. So measuring strain A in plasma might, down the line, give us a clue about an individual's risk for faster cognitive decline in Parkinson's disease. That could be incredibly valuable for managing expectations and planning care. But there's still this fundamental question. Are these just markers, or are they actually involved in the disease process? Do these alpha-synuclein strains floating in the blood do anything harmful?
And a critical question, and they tackled it directly. They used those seed amplification assays I mentioned earlier. They took the alpha-synuclein species isolated from plasma using the strain A and strain B antibodies and tested if they could seed or trigger the aggregation of normal alpha-synuclein monomers, and they could. Both strains showed seeding activity, but the species isolated with the strain A antibody seemed to be more potent seeds. Even more specifically, when they compared the strain A species isolated from plasma of Parkinson's patients versus dementia with Lewy bodies patients or normal controls, the strain A material from the Parkinson's disease plasma induced more alpha-synuclein and fibrillization in the assay. It had a stronger seeding capacity. And they went a step further, using a cell culture model. They used SK-MLL28 cells, which can be induced to form alpha-synuclein aggregates. When they applied the strain A alpha-synuclein isolated from Parkinson's disease plasma to these cells, it triggered the formation of those characteristic detergent-insoluble alpha-synuclein clumps. This effect wasn't seen when they used strain B material or material isolated with antibodies against total alpha-synuclein.
Wow, so it's not just an association. The strain A alpha-synuclein from Parkinson's patient plasma actually seems to have the biological capability to drive the aggregation process, at least in these lab models. That really elevates its potential significance beyond just being a marker.
Exactly. It provides pretty compelling evidence, albeit from in vitro and cell-based studies for now, that these plasma alpha-synuclein species, especially strain A in Parkinson's disease, possess real pathogenic potential. And zooming out, these findings really bolster this strain hypothesis for Lewy body diseases, the idea that different shapes of alpha-synuclein lead to different biological effects and ultimately different clinical diseases. Seeing this play out with human samples is a significant step.
It really shifts the perspective, doesn't it? The fact that these informative, potentially pathogenic strains are enriched in plasma, not clearly in cerebrospinal fluid, and don't directly track with brain levels. It strongly suggests we need to look beyond the central nervous system. Could the primary source, or at least a major contributing source, be peripheral, like the gut maybe, or the autonomic nervous system?
That's the million-dollar question this raises. It definitely fuels the discussion around the gut-brain axis or other peripheral origins in Parkinson's disease pathogenesis. If these strains are originating or accumulating peripherally and perhaps influencing the central nervous system from there, or even representing a distinct systemic aspect of the disease, well, that changes how we think about disease initiation and progression. It implies that focusing solely on the brain might miss crucial parts of the story.
And the therapeutic implications are potentially enormous. If pathology starts or is significantly driven from outside the central nervous system, maybe we could target it there before it even reaches the brain or alongside brain-targeted therapies.
Precisely. It opens up possibilities for peripheral therapeutic targets. And in terms of clinical trials, these blood-based conformational biomarkers could be incredibly useful. They might complement cerebrospinal fluid seed amplification assays, perhaps giving different but related information. Using both could allow for much better patient stratification, identifying the right patients for the right trials based on their specific biological profile. The robustness of these findings across multiple centers really adds weight to this potential. Of course, we need to be balanced. There are limitations. We need in vivo studies to confirm the pathogenicity of these plasma strains in a living organism. And we still need to pin down exactly where they come from. And yes, there was some variability between the different study sites in the larger cohort that's common in multi-center studies. But the core finding, the ability to differentiate Parkinson's from dementia with Lewy bodies, held up. That's the key signal here.
Okay, let's quickly recap the practical takeaways for you, our listeners. This study identifies specific alpha-synuclein strains in blood plasma that show promise for distinguishing Parkinson's disease from dementia with Lewy bodies. Plasma levels of strain A and strain B were found to be significantly higher in Parkinson's disease patients across two large groups. Critically, higher baseline plasma strain A was linked to slower future cognitive decline in those with Parkinson's. And importantly, these plasma strains, especially strain A from Parkinson's patients, showed they could actually seed alpha-synuclein aggregation in lab tests. So this opens exciting paths towards blood tests for diagnosis, cognitive prognosis, and potentially gives us new insights into disease mechanisms that might extend beyond the brain. And perhaps the most provocative thought to leave you with is this. How much does pathology outside the central nervous system truly shape these neurodegenerative diseases? If these potent alpha-synuclein strains have significant origins or reservoirs outside the brain, what does that fundamentally mean for how we should be trying to treat or even prevent these devastating conditions? Thank you for joining us on this weekly Neurology Deep Dive, a production by America Venini. We strongly encourage you to read the full paper in the Annals of Neurology to explore these important findings in greater detail.